Very good morning, students. The today topic is functions of liver. The last class I told about the structure of liver. Okay, liver is the largest gland consisting two lobes. Each lobe of liver having several lobules called aortic lobules. These aortic lobules are the structural and functional unit of the liver. Coming to here, before to that one, the structure of liver having many lobules called aortic lobules. Each lobule have several hepatocytes or liver cells are there. The liver cells are hepatocytes which are arranged in the form of cord-like structure or chain-like structure is called hepatic cord. These hepatic cords which are arranged like a radial manner around the central vein. Now each hepatic lobule it is appear hexagonal in structure. On its surface is enveloped by the thin connective tissue membrane called Glissen's capsule. From the hepatocytes, here bile juice is secreted. The secreted bile juice is received by the hepatic ducts. These hepatic ducts, which are organized to form the liver ducts. Hepatic ducts are organized to form liver duct. The liver duct, which receive the bile juice from the liver and released into gallbladder. Gallbladder it helps in storage the bile juice and concentration of bile juice. This concentrated and stored bile juice is received by the cystic duct. Now this is a cystic duct. The cystic duct is united with the liver duct. These two ducts are organized to form common bile duct. See, this is a common bile duct. The common bile duct it is formed by the duct of liver and cystic duct or duct from the gallbladder. See, this is the gallbladder. From the gallbladder, duct is arise. It is merged with the duct from the liver. These two ducts are organized to form or united to form common bile duct. Now, this common bile duct it is merged with the pancreatic duct here. Common bile duct it merged with the pancreatic duct. These two ducts are united to form hepatopancreatic duct here. This is the pancreas. Now from the pancreas, pancreatic juice is received by the pancreatic duct. This pancreatic duct it is merged with the common bile duct. Here these two are united to form here hepatopancreatic duct. Now this duct it is opens into duodenum. This duct opens into duodenum by the sphincter of Oddi. Sphincter of Oddi it is the sphincter wall. Here it is opens between the it is present between the duodenum and hepatopancreatic duct. Where the hepatopancreatic duct is opens into duodenum, in that area is guarded by sphincter of Oddi. So other name of the hepatopancreatic duct is called ampoule of water. Ampoule of water. Hepatic duct from the hepatic duct from the liver form the common bile duct. The CBD, that is common bile duct, is joined by the pancreatic duct to form ampoule of water. Ampoule of water nothing but a hepatopancreatic duct here. Here is hepatopancreatic duct or ampoule of water. It opening into the duodenum is guarded by the sphincter of Oddi. You should remember, students, this is important to you. This also very important. Here ampoule of water. Say ampoule of water, other name hepatopancreatic duct here. Ampoule of water is also called hepatopancreatic duct. This duct is made by the duct from the gallbladder and liver and duct from the pancreas. These two ducts are organized to form or united to form hepatopancreatic duct. So other name of this one is called ampoule of water. It opens into duodenum. It opens into duodenum. Duodenum is guarded by the sphincter of Oddi. Now coming to here functions of liver. Functions of liver. The liver it play several roles in our body. So because of it, the largest organ in our body. Here the liver it shows following three main functions. One is synthesis, second one is storage, and other one is secretion. Synthesis, storage, secretion. The three main functions are showing by the or performed by the liver. Now Coming to what type of substances are synthesized, what type of substances are stored, and what type of substances are secreted by the liver. Let us coming to the first function of the liver secretion. It secrete here bile juice. Bile juice here it is secreted by the liver cells. Now it is appear yellowish green in color. It contains basically two components. One is sodium. Yeah, it contains in two components such as glycocholates and tarocholates. These are the actually salts which are present in the bile juice. No enzymes in the bile juice, though it not contain any enzymes here. It play important role in the metabolism. The bile juice contain basically two salts and two pigments. What are the two salts? Glycocholate and tarocholates of sodium and potassium, and bilirubin, bilirubin. These bilirubin, they give the color to our fecus and urine. They give the color to our fecus and urine. Next, glycocholate and tarocholates of sodium and potassium, they were helps in the emulsification of fats. Fats emulsification is performed by the salts of bile juice. What are the salts of bile juice? Glycocholates and tarocholates of the sodium and potassium helps in the emulsification of fats. Emulsification means a large sized fat globules which are converted into simple fat droplets called emulsification. That process is completed by the glycocholates and tarocholates of sodium and potassium. Now coming to bile pigments, bilirubin, biliverdin. Actually, these bilirubin, biliverdin, these two are formed by the degradation of the hemoglobin, the RBC. Whenever our RBC lifespan is 120 days, after 120 days, they goes to liver and rested within the liver cell. Therefore, the hemoglobin is disposed. This disposing hemoglobin here or breakdowning hemoglobin, it digested by the body and produces bile pigments. They are bilirubin and biliverdin. Simply, bile pigments are bilirubin, biliverdin. They are the representation of the dead RBCs in our liver. And coming to next function, that the liver play important role in the metabolism of carbohydrates, metabolism of the lipids, and metabolism of the proteins. Here carbohydrate metabolism, in the case of here glycogenesis, glycogenolysis, glucogenesis, and lipogenesis. Glycogenesis means production. Glycogen is produced from the glucose. If our body have excess glucose is there, that excess glucose is immediately conversion to glycogen is called glycogenesis. It occurs in the liver. Like that, glycogenolysis. If our body glucose level is very less, so immediately the stored glucose is conversion to glucose is called glycogenolysis. Lysis means splitting. What is splitted? Glycogen is splitted into glucose called glycogenolysis. Glycogen conversion to glucose called glycogenesis. Here glycogen is generated called glycogenesis. Glycogen is splitted is called glycogenolysis. And gluconeogenesis. Gluconeogenesis means if the glucose is coming from the non-carbohydrates such as other than starch, cellulose, glycogen, etc. Except carbohydrates, if the glucose coming from the non-carbohydrates is called gluconeogenesis. And lipogenesis means the production of the glucose from the lipids is called lipogenesis. So these are the comes under carbohydrate metabolism of the liver. And coming to your liver also play important role in the lipid metabolism. Lipid metabolism means how the lipids are synthesized in our body here by the process of here the breakdown of the the synthesis of cholesterol and production of the triglycerides. Cholesterol nothing but fat. The fat is produced here whenever the produced fat is synthesized here finally conversion to triglycerides, diglycerides and monoglycerides. These are formed, these are synthesized take place in the liver. That is called lipid metabolism, and followed by the protein metabolism, especially here the formation of ammonia, the waste material. Here it is highly toxic substances. Here if it is remains in our body, here immediately here that conversion to poison or toxic substances, the toxic agent, it is immediately conversion to here urea and uric acid by the process of deamination. The first one, deamination means the removal of amino group from the amino acid. Amino acid is the simple form of the proteins. Here it contains a NH2 group and COOH group. That NH2 group is removed from the amino acid is called deamination. After removal of the amino group, that amino group is removed and joined with the H plus ion, means hydrogen ions. Here make the NH3. That is called formation of ammonia. For formation of ammonia require amino group. That amino group is removed from the amino acid is called deamination. Now that ammonia is joined with the carbon dioxide make urea via Arndt cycle or urea cycle. Arndt cycle or urea cycle. So it is the metabolic cycle. This metabolic cycle, it is useful for formation of urea. Urea is the another nitrogenous waste metabolism. That nitrogen waste metabolism is formed by the union of carbon dioxide and ammonia through the Arndt cycle or urea cycle. Next one, and also liver also helps in the formation of glucose. Formation of glucose from the lactic acid. So with the lactic acid, have you seen in our body skeletal muscles are the fatigue. Why the skeletal muscles are fatigue? If the continuous contraction of the skeletal muscle, our muscle glucose become to disappear. So due to continuous contraction of skeletal muscles. So why the glucose disappear? For the contraction of muscle require a lot of energy. That energy is obtaining from the glucose via anaerobic respiration. Therefore, here through the anaerobic respiration, here ATP and what is formed? Lactic acid is formed. Lactic acid is the end product of the anaerobic respiration in the skeletal muscle and other cells in our body. So, here due to anaerobic respiration from the glucose, first one what is formed? Lactic acid is formed. Now that lactic acid is conversion to glucose. That lactic acid is conversion to glucose. Via pyruvates takes place in the liver is called Cori cycle. Cori cycle is the cycle refers to conversion of glucose from the lactic acid. Glucose from the lactic acid form takes place in the liver is called liver by Cori cycle. The process of conversion of glucose from the lactic acid is called gluconeogenesis. Just now I told about the gluconeogenesis. If the glucose is coming from the non-carbohydrate, lactic acid is the non-carbohydrate. Actually, it is the end product of the glycolysis. Which glycolysis here? Anaerobic glycolysis. Through the anaerobic glycolysis here, lactic acid is formed. The lactic acid is conversion to glucose through the gluconeogenesis here. And next one, here liver also is the chief organ for detoxification. Detoxification means if our ingesting food material contain any toxic substances are there, that's are toxic substances which are conversion to non-toxic substances is called detoxification. Detoxification means here toxic substances are toxic conversion to non-toxic or detoxic is called detoxification. The this box is very important to you. Liver is the first checkpost that bears the entry of what are that toxins into general circulation. Recall the role of smooth ER. You know about the ER. ER is the endoplasmic reticulum. ER have two forms. One is smooth and another one rough. The smooth ER it play important role for the detoxification, lipid metabolism. Now liver contain large amount of the smooth ER. Therefore liver play important role in detoxification. Alcoholic person means those who are addicted to alcohol. In those persons here their liver is affected with the liver cirrhosis. Liver cirrhosis means here alcohol addicted person here in its liver is infected with some fungal toxins such as Aspergillus flavus. Aspergillus flavus is the fungal toxins. Here it damage the liver. Therefore the liver become to non-functioning. Here that is called liver cirrhosis. Here the why the liver cirrhosis occur in the alcoholic person. In alcoholic person liver function is very poor. So due to destroy the liver cells in the liver by the alcohol, and here in alcoholic person liver is damaged or liver is affected. Here after entering the body, the aflatoxins are generally metabolized by the liver into some other non-toxic or less toxic substances. If tolerate the liver, there is no problem to liver. If you cross its tolerate level, your liver become to or undergoes here damaged here. This is the specialization of liver. You should remember liver contain large amount of smooth ER, hence it act as a detoxified organ. Should remember, very important. Now coming to liver also act as a thermoregulatory organ. Thermoregulatory organ means here our body temperature is regulated along with the muscles and brain hypothalamus. Here along with the muscles and hypothalamus our body temperature is regulated by the liver. Therefore liver is the thermoregulatory organ, means it is a part of thermogenesis. Liver also act as a hemopoietic organ. Hemopoietic means hemopiesis means the process of production of blood cells is called hemopiesis. Hemopoietic organ means here liver it helps in the production of RBCs before birth, especially at fetal stage. Whenever after birth here it act as agonist function to hemopoietic organ, means erythroclastic organ. Erythroclastic organ means after birth here liver it helps in the engulf the dead RBC cells along with the spleen. After birth the liver act as a erythroclastic organ. Erythroclastic means engulf the dead RBC cells here. Here hemopoietic organ means before birth or at fetal stage. Liver act as a hemopoietic organ. It means production of RBC from the liver before birth, but after birth liver it act as a erythroclastic organ, means engulf the RBC cells, dead RBC cells. And liver also it helps in the synthesize the some proteins such as albumin. It is abundant protein in our blood, and globulin. Next one also produce the fibrinogen, prothrombin, etc. Albumin, globulin, these are the main blood proteins in our blood plasma, and they were helps in the maintain the acid base buffer in our liver, in our blood. And coming to here, our blood clotting protein is also synthesized by liver, such as fibrinogen, prothrombin, and also it produces heparin. Heparin is the anticoagulant. These two are the coagulants. These two are act as acid base buffers to our blood here. So these 5 proteins are synthesized by liver. Next one, liver act as a liver cells. Liver also contain lot of Kupffer cells here. Kupffer cells means here they are the modified phagocytic cells in our liver. Whenever here microbial organisms or unwanted cells or dead cells are dead cells are circulated to liver. These circulated are received unwanted microbial organism, received unwanted cells are dead cells or microbes which are engulfed by the specialized phagocyte cells in the liver. They are called Kupffer cells. Kupffer cells here. They are present in the sinusoids that in between the hepatic cord. So what are the sinusoids? Sinusoids means the place occurs between the place occurs between the hepatic cords. See, this is the hepatic globule, this is the one hepatic cord, this is also another hepatic cord. Between any two hepatic cords it has space is there. That space is called sinusoids. In it here, so Kupffer cells are there. Kupffer cells which are helps in here engulfing of the dead cells or engulfing of the microbial organisms. And coming to next one, another gland in the digestive glands, that is pancreas. Pancreas is also known as mixed gland, or is also known as your heterocrine gland. Mixed gland or heterocrine gland here is also sweet gland. So because of here the glucagon hormone is secreted by this gland, hence it is also known as sweet gland or mixed gland or heterocrine gland. Mixed gland means here it secretes enzymes as well as hormones. Heterocrine function means it acts as endocrine as well as exocrine. Exocrine and endocrine means either act as a duct gland or ductless gland. Duct gland or ductless gland. Therefore, it is an heterocrine gland. And coming to where is it found? Pancreas located. Our pancreas located here limbs of duodenum of small intestine. This is the limbs of duodenum of small intestine. See, this is the small intestine, duodenum part of small intestine. So in this area, what is there? Pancreas is there. The U-shape or C-shaped duodenum, C-shape or U-shaped duodenum of small intestine. Here it has two limbs. This is one limb, the another limb. Between two limbs here the leaf-like elongated gland is there, is called pancreas. It is a mixed gland or heterocrine gland or sweet gland. Now coming to pancreas, here is the second largest gland in human body. Here having two types of cells here in the liver. Say the peripheral region of the pancreas containing here chief cells or SNA cells here. Peripheral region of the pancreas contains here chief cells or SNA cells, whereas the middle part of pancreas having some endocrine cells. The group of the endocrine cells present in the pancreas is called islets of Langerhans. They secrete insulin, glucagon and other hormones here. Pancreatic juice here contain how does secrete the how secrete the pancreatic juice here? Pancreatic juice secreted by the exocrine cells in the pancreas, whereas the hormones are secreted by the endocrine cells in the pancreas. What are the exocrine cells in the pancreas? Chief or SNA cells. They are called pancreatic acini. Pancreatic acini secretes pancreatic juice, which contains enzymes mostly. Whereas the middle of the pancreas contain endocrine cells. The group of the endocrine cells in the pancreas called islets of Langerhans, which secrete insulin, glucagon and other hormones here. Now pancreatic juice which contain following components. They are sodium bicarbonate, next one trypsinogen, chymotrypsinogen, carboxypeptidases, pancreatic lipase, pancreatic amylase, pancreatic lipase, carboxypeptidases, and chymotrypsinogen, trypsinogen, sodium bicarbonate. Along with them also have nucleases such as DNAs and RNAs. So these are the components which are present in the pancreatic juice. So this is the pancreas. These are the digestive glands. Now let us come to here physiology of digestion. Physiology of digestion means how our ingesting food is digested in our alimentary canal or in our digestive system. That is called physiology of digestion. What is the digestion? Digestion is the process in which your conversion of complex non-diffusible food substances into simple diffusible forms. Non-diffusible substances means unable to absorbing the food material into blood. So due to they are large in size here. Before digestion, our ingesting food particles are very large in size. They are unable to absorbing into blood away from the intestine. So therefore, these non-diffusible substances which are conversion to simple diffusible forms is called digestion. The digestion process is accomplished by the mechanical digestion and chemical digestion. Mechanical digestion means cutting and chewing the food by the teeth and churning the food by the peristalsis here. Cutting and chewing the food by the teeth means our food is ingested. If the food is ingested into buccal cavity, that ingested food is cutted and chewed by the teeth, and later they are churning. Churning means mixing the food with the help of peristalsis movement. That is the mechanical digestion. Churning the food takes place in the mouth and in stomach, whereas chewing and cutting the food takes place in the mouth. Now here mechanical digestion followed by the next digestion is called chemical digestion. Chemical digestion means while the food is after cutting and chewing the food, the cutted and chewed food is exposed to some chemical components in the buccal cavity as well as in the other part of alimentary canal. Yeah, with the help of some enzymes, that is enzymatic action here or enzymatic reaction occurs in the alimentary canal is called chemical reaction. So these two processes are occur in the physiology of digestion. One is mechanical digestion, another one is chemical digestion. Remaining topic I will explain tomorrow class about the process of digestion in buccal cavity, process of digestion in stomach, small intestine. Thank you.